Hello, hope I'm audible and I hope my presentation is also visible. Yeah, so this is the last session of mine for this course, week 12, Experimental Biotechnology by Professor Vishal Trivedi from IIT Guwahati. So I'm one of your TAs, Sumati from University of Hyderabad. So since this is the last session, and it's for the last week. It's more of a revision and summary and conclusion sort of questions that were given in your assignment. So let's go by the questions one by one and just discuss the topics in brief. So the first one was regarding the OECD guidelines for testing of chemicals. So this was in the year 1981. The OECD is the Organization for Economic Cooperation and Development, and it is an international body that promotes policies for economic growth, environmental protection, and social well-being. So in 1981, OECD officially published the guidelines for testing chemicals, which became internationally accepted, and these are the standards that are being followed or should be followed for chemical testing. So the purpose of the guidelines are, the main objective was to standardize test methods that are used to evaluate the safety and environmental impact of chemicals, then facilitate international acceptance of test results, avoiding repetitive testing in different countries, and then support regulatory decisions regarding human health and environmental safety. These guidelines, however, uh, cover a wide range of testing areas like toxicology, ecotoxicology, physicochemical properties, environmental fate and behavior, etc. So, in toxicology, again, we have many things like acute toxicity, chronic toxicity, reproductive, developmental toxicity, etc. And in ecotoxicology, it is the effects on the aquatic and terrestrial organisms, and physical chemical properties like solubility, partition coefficient, vapor pressure, etc. of the chemicals come under these. And environmental fate and behavior is the biodegradation of the chemical and bioaccumulation of the chemical. So all these are under test and are maintained by the standards of the guidelines here. And there is something called as mutual acceptance of data, that is the MAD principle. So alongside the guidelines, OECD also introduced the MAD principle. This means that the data generated in one OECD member country in accordance with the OECD test guidelines and good lab practice shall be accepted by other countries for assessment purpose. So, like we said in the previous, it is internationally accepted. So, it is mainly promotes that the testing that have been done in one country is internationally accepted, and there is no need of repetitive testing. So this drastically reduced duplication of experiments, saved resources, and minimized animal testing while ensuring high quality and reliability in the data across regulatory systems. So, what is the impact of this? Is the OECD test guidelines are now recognized worldwide, and the frequency and it is frequently updated to reflect new scientific advances. They form foundation of international chemical safety assessment programs, the REACH in EU, that is in Europe, and EPA in US, and Also, their use promotes harmonization, transparency, and efficiency in the testing and regulation of all the chemicals that are being used. So, therefore, the OECD guidelines are widely accepted worldwide for testing of chemicals, and these were published in the year 1981, and they represent a landmark step in creating an internationally harmonized system for evaluating safety of chemicals, ensuring the test results are reliable, reproducible, and mutually accepted among countries. So this was about the OECD guidelines. Next is regarding the Lyme disease and the biosafety level of it. So, Lyme disease is caused by bacteria. So, the causative agent is Borrelia burgdorferi, and it's a spirochete bacterium. The vector is Ixodes ticks, so commonly called as deer ticks or black-legged ticks, and the transmission is through the bite of the infected ticks and not through air or causal contact. So it is via the bite of the infected tick that it transfers and transmits. Then the disease is zoonotic type, and it's a vector-borne bacterial infection. Lyme disease affects the skin, joints, nervous system, and the heart, but it is generally treatable with antibiotics like doxycycline or amoxicillin, and it is definitely in BSL-2. BSL-2 is designated for agents that can cause moderate human disease, pose limited risk of transmission, and have effective treatment and preventive measures that are available. So, since Borrelia burgdorferi does not spread like through air, it's not an aerosol. So, uh, the diseases or like the viruses or anything which spread through air or contaminated environment are in BSL-3 facilities. So since this is zoonotic mode of transmission, it's only through the bite of the infected tick it transmits. So it's not placed under BSL-3, it is under BSL-2. So it is not usually fatal, and it is treatable, and the appropriate biosafety contamination level for laboratory work is BSL-2. The key BSL-2 laboratory practices for Borrelia burgdorferi. So BSL-2 basically is also for pathogens, so their access is restricted. Only authorized personnel may enter the laboratory during experiments, and PPE is mandatory, lab coat or gown, and gloves, which are changed frequently, face and eye protection when splashes or sprays are possible, and containment equipment procedures with potential for aerosol generation, example like during centrifugation and vertexing, pipetting, etc. All these should be performed in class 2 biological safety cabinet. So it's generally a airflow laminar with HEPA filters in it. Then there is disinfection and waste disposal. So this is most important when you're working with pathogens and live organisms or live cells. So work surfaces must be disinfected with agents against bacteria like 70% ethanol or 10% bleach, and autoclave all the biohazardous waste before disposal. Hand hygiene is important. Wash hands after infectious uh, materials are handled and also before leaving the lab. And then training is a must. It must all the personnel who are authorized must be trained beforehand, and the handling of the pathogenic microorganisms and also emergency procedures should be taught to everybody who handle or who have access to this BSL-2 facility. So, as we said, why BSL-2? Because it's pathogenic. BSL-1 is usually non-pathogenic organisms, so it is not placed under BSL-1. And since it is not aerosol or it's not untreatable, it's not placed under BSL-3 or 4. So, since it's pathogenic and it has efficient treatment, it is under BSL-2. BSL-1 is used for non-pathogenic organisms like E. coli, etc. And Borrelia burgdorferi is pathogenic and is too risky in BSL-1 facility. And like I said, for BSL-3, it is used for airborne or serious pathogens like Mycobacterium tuberculosis. And Lyme disease is not airborne, so BSL-3 is excessive and not required. BSL-4 is for highly lethal, untreatable agents like viruses, like Ebola. So, Lyme disease is treatable and not fatal, so it is not BSL-4. So, this is a brief about the disease. The pathogen is Borrelia burgdorferi and it's called as Lyme disease. Transmission is through zoonotic infected tick, and it is, even though not highly infectious since it's not airborne, it is, however, moderately risky to the lab workers. So the facility must be maintained very well and disposal should be done very efficiently. Availability of treatment is there and antibiotics are used for it, and it's BSL-2, and the reason is it's moderately hazardous, treatable, no aerosol transmission. So that is about the Borrelia. And the next was regarding SDS page and what is the pH of the running buffer. So in SDS page, which is polyacrylamide gel electrophoresis with detergent SDS. Several buffers are used at different stages. So we use buffers for preparation of the stacking gel, resolving gel, and also for running the samples during the electrophoresis. So for running buffer, which is basically made up of trisglycine and SDS, the concentration is 25 millimolar trisbase and 192 millimolar glycine and 0.1% SDS. And the pH of this buffer is usually 8.3, and it is commonly referred to as trisglycine SDS buffer or SDS running buffer. The function of this is it provides ions that carry current during the electrophoresis. So it helps maintain a constant pH, so the proteins migrate properly according to their size, and that is they resolve according to their molecular weight. And SDS ensures that the proteins remain denatured and uniformly negatively charged. So the pH of the running buffer in SDS page is 8.3, which maintains proper protein migration and also ensures efficient electrophoretic separation. So this was about the running buffer. And other than the running buffer, as I said, we also have buffers which are used to make the stacking gel and the resolving gel. So these are generally tris buffer, tris HCl buffer. So these are made in two pHs. One is 8.8 and the other is 6.8. So the molarity also is different. One is 1 molar and the other one is 1.5 molar. So the stacking gel is made with a tris buffer of 6.8 pH, and resolving gel is made with the pH of 8.8. The buffer used for stacking gel is to stack the proteins into sharp bands before entering the resolving gel. So they have to run form a nice band to run evenly on the gel. And then resolving gel is basically to separate the proteins based on their size. This is the main gel for separation, and the running buffer is the trisglycine SDS buffer, and the pH is 8.3, and it's used to maintain ionic strength, conductivity during the electrophoresis. Next is ion exchange chromatography. So, in ion exchange chromatography, it's classified whether based on the exchanges. So, if it's like a cationic or anionic, that is based on charge, and if it's based on the strength of the ionic groups, we have another chromatography. So ion exchanges, that is the anion exchanges, they bind negatively charged ions, and therefore the exchanger must be positively charged. The positively charged functional groups, so they bind on the negatively charged anions. So in case of strong anionic exchanger, we have sacs, and the weak anion exchanger wax. So the functional group differs, and for a strong anionic exchanger we have quaternary amines, and for weak we have DEAE, and strong are fully ionized across all pH values, whereas the weak anion exchanges are partially ionized, and it depends on the pH, uh, whether the strength of it varies, etc. So the examples for strong anion exchanges are Cephirose, Q-Cephirose, QAE, Cephidex, etc. And for weak exchanger, we generally use DEAE, cellulose. So why quaternary amine is a strong anion exchanger? So quaternary amines have a permanent positive charge on the nitrogen of the four alkyl groups attached. So quaternary is basically having four alkyl groups, and this makes them ionized over the time, over the entire pH range. Like if there is even a change in the pH, slightly up and down, the ionic strength is still very, very strong. It doesn't change for a very large range, and hence a strong anion exchanger. So they bind strongly to the anionic species such as phosphate, sulfate, or carboxylate ions. So this is a quick table that was given. So sulfonic acid is a cationic exchanger, and it is a strong cationic exchanger. And carboxylic acid is a cationic exchanger, but it's a weak cationic exchanger because its ionization depends on the right pH. And quaternary amine is an anionic exchanger, and it's very strong because it is very stable in a varied range of pH. And the EAE is an anionic exchanger which is weak, and it also partially ionized depending on the pH. So therefore quaternary amine is a strong anion exchanger and is fully ionized over all pH ranges. Next is regarding the isoelectric point. So what exactly is isoelectric point? A, it is a point of a protein that is at a pH which when the protein carries no net electrical charge. So the pH at which there is no uh, net electric charge on a protein is called the isoelectric point of that protein. So at this pH, the positive and negative charges in the amino acid residues balance out each other. So therefore the net charge is zero. So pH is equal to Pi, therefore the net charge becomes zero. And how it affects the solubility is it decreases it, and if the pH is far from Pi, the protein carries either a net positive or a net negative charge, and it increases the solubility of the protein. So at a pH near Pi, there is no electrostatic repulsion between the protein molecules because they have no net charge on it. The net charge on it is zero. So there will be no forces of electrostatic repulsion. This lack of repulsive forces allows the protein to aggregate and precipitate, and hence the solubility is minimal. When the pH values move away from the pi, then the protein carries a net charge. So it's either a net positive or a net negative. This leads to electrostatic repulsions and it prevents aggregation of the protein. So thereby it increases the solubility of it as well. So that was about the PI. So for example, the PI of bovine serum albumin, that is BSA, is 4.7. So solubility of BSA is lowest at 4.7 pH. The solubility increases both at acidic and basic pH, that is when the pH of your solution is below 4 or above 5. In both the cases, the solubility of BSA increases, but around at 4.7 it will precipitate. So proteins have minimal solubility at pH values near their isoelectric point because they carry no net charge, reducing the electrostatic repulsion and promoting the aggregation and precipitation. So next is regarding the affinity chromatography. So in affinity chromatography, separation is based on specific biological interactions. So it is between a molecule, a ligand molecule that is attached to the stationary phase and its complementary binding partner which is present in the sample, which could be either a receptor or a target protein. So basically it is the interaction between a ligand and a receptor molecule or a target molecule. So the ligand with affinity for lysine, uh, the lysine residues have the primary amine group, and a commonly used affinity ligand that interacts with such amino groups is Sebacron blue, and it is also known as blue sephirose. So it's Sebacron blue F3GA. So it mimics the nucleotide cofactors like NAD plus, NADP plus, and FAD. So therefore, it binds strongly to the NAD plus dependent enzyme. So enzymes usually need several cofactors. So one such cofactors are the nucleotide cofactors, which are NAD, NADP plus, and FADs. So since the Sebacron blue mimics the structure of this, it can bind to the enzymes which usually bind to the nucleotide cofactors. So enzymes like dehydrogenases, kinases, and also some serum albumins usually depend on these NADs as cofactors. So they have a binding site for it. So background blue mimics this nucleotide cofactor and binds to these NAD plus dependent enzymes. So the binding often occurs via lysine residues near the coenzyme binding site. So the relationship between the lysine and the NAD plus dependent enzymes, the epsilon amino group of lysine can form ionic or hydrogen bonds with the diligand, that is the Sebacron blue ligand. Then NAD plus containing dehydrogenases have specific structural regions, that is, they have the active sites that include the lysine residues which are involved in the cofactor binding. Therefore, they show strong affinity for matrices carrying the diligands and interact with the lysine. So NAD plus containing dehydrogenase, because in affinity chromatography, diligands like Sebacron blue F3GA bind to the lysine rich region. So this is present in the NAD plus dependent enzymes. So this is exploited and used for their cofactor binding affinity in case of affinity chromatography. Next is regarding Franz adjuvant. So it is a substance that is used to enhance the immune response of an antigen. So we know there is something called as an immunogen and an hapten, carrier, adjuvant, etc. So Franz adjuvant is basically of two forms. So there is incomplete Franz adjuvant and there is complete Franz adjuvant. So the complete Franz adjuvant composition is made up of mineral oil, emulsifying agents such as mannite monoileate, and heat killed Mycobacterium tuberculosis. Excuse me. So, mineral oil acts as an emulsion medium, and emulsifying agents like mannite is added, and in some cases, heat killed Mycobacterium tuberculosis is used. So, the function of Mycobacterium tuberculosis is the bacterial cell wall components of it have substances like mycolic acids and muramyl dipeptides, and these are strong immunostimulants. So they activate the macrophages and the dendritic cells, leading to enhanced antigen presentation and a robust cell mediated immune response. So this comes under complete Franz adjuvant. Then there is incomplete Franz adjuvant. So it contains only mineral oil and emulsifier and does not have mycobacterium. So when it is used when a milder immune response is desired or for a booster immunization. So this gives a complete stimulation of the immune response. And this one, however, since it does not have mycobacterium, it is to either boost the previous immunization or to invoke a milder response. So, this is a table about the nature of Franz complete and incomplete adjuvants. So, in your complete, we have the heat killed Mycobacterium tuberculosis. In the incomplete, we do not have it. And the type of immune response of complete is strong, cell mediated, and humoral response. Incomplete is, however, mostly humoral response. Then complete is primary immunization and incomplete is booster injections. So, however, complete Franz adjuvants induces strong inflammation, and it is not advised to use for humans, and it is only in experimental animals, and it also has severe local tissue reaction as well. So, complete Franz adjuvant consists of the heat killed Mycobacterium tuberculosis and provokes a strong, complete cell mediated and humoral response when immunized. Then, next is the clumping of red blood cells by viral particles. And clumping is basically like aggregation, and when this is taking place due to viral particles, it is known as agglutination. And since it is of RBC, it is called hemagglutination. So basically, agglutination is when the particulate antigens such as cells, bacteria, or viruses are clumped together in the presence of specific antibodies or other binding molecules. It involves cross linking between the particles forming a visible aggregate. So, when there is antigen and antibody complex formation, so these tend to agglutinate. Like also in case of blood test, when it is done, you when you add your blood and you add the antibody against your specific type of antigen present in your RBC, you see agglutination. So based on the agglutination, we decide whether that specific antigen is present on your RBC or not. So in the same way, this is also uh, the clumping. This is because of the clumping, and it's called as hemagglutination. So this is the cross linking between the particles which forms visible aggregates. And then we have viral hemagglutination. So this also occurs when certain virus particles are present, then like influenza, measles virus, mumps virus, etc. They possess surface proteins which is called as hemagglutinin. So this can bind to the sialic acid receptors on the surface of red blood cells. So this binding causes clumping of RBCs and a visible lattice formation is seen. So when they clump with each other, they tend to form like a nice organized lattice, and this is called as hemagglutination. And this is used in hemagglutination assays as well. So the example for such viruses are influenza virus hemagglutinin HA. It binds to the sialic acid residues on the RBCs, and then RBCs clump together and agglutinate. Then, so there are different uh, terms that are used which are generally confused. So there is a slight difference between each other, even though everything is inadvertently an aggregation process. So precipitation, agglutination, immunodiffusion, immunoprecipitation. So precipitation is when formation of an insoluble complex between a soluble antigen and a antibody occurs. So here the antigen is a soluble antigen, and excuse me, and in case of agglutination, clumping of the insoluble particles or cells by specific interaction takes place. So here you have a particulate matter, and when this forms interactions and forms a lattice, it agglutinates. So example in case of RBCs, bacteria, etc. So in case of precipitation, you have a soluble antigen. When it forms bond with the antibody, then the complexes form and it precipitates. But in case of agglutination, we have a particulate matter which will start clumping due to specific interactions and will result in agglutination. Then comes immunodiffusion. So, in case of immunodiffusion, antigen and antibody diffuse into the agar to form a visible precipitin line. So, here also the antigen is soluble. So, this is an assay. So, it is either the octolunate diffusion or immunodiffusion, where you we have discussed this earlier, I guess. So, we have an agar plate, and then we punch holes in the agar and we add our antigen in it, and then we have our antibody in our agar. So at a particular place, when there is a zone of equivalence, we have a precipitin ring formation. So this is called as immunodiffusion. Then immunoprecipitation. So precipitation is used for protein isolation in molecular biology methods. So here we need to precipitate out the protein, the isolated protein. So we use antibodies and we pull it down. So this is called as immunoprecipitation. And here also your protein is soluble, and when it is exposed to the antibody, it will bind to it, form complexes, and it will precipitate out. Then, what is the diagnostic importance? So, hemagglutination assays are used to detect or quantify viruses that can agglutinate RBCs, and hemagglutination inhibition test is used to detect antibodies against these viruses and the antibodies that can prevent the agglutination. So, the clumping of the red blood cells by viral particles is an example of agglutination, specifically hemagglutination, because it is the RBCs that are getting agglutinated. So next is an assertion and reason based question. So the assertion is trypan blue dye contains only trypan blue dye stains only the dead cells, and the reason given is trypan blue is a charged dye and it cannot enter the live cells due to the presence of membrane potential. So here the assertion is true, that is the trypan blue stains only the dead cells. Uh, The trypan blue is a vital dye and it is used for cell viability assays. And the live cells exclude the dye, while the dead cells with compromised membranes allow the dye to enter and stain the cytoplasm. But however, the reason is not completely true. The trypan blue cannot enter the live cells, that is true, but the reason given that it is due to the membrane potential is incorrect. So it is due to the pore formation or due to the uh, laws of integrity of the membrane itself that allows the trypan blue to enter into the cells. That is, the exclusion of trypan blue from live cells is not due to membrane potential but due to the intact plasma membrane. Live cells maintain a selectively permeable membrane that prevents large or charged molecules like trypan blue from entering in. Once the membrane is damaged in the dead cells, the dye freely enters and stains the cytoplasm. So that is how we can visualize the dead cells and differentiate them from the live cells. Next is regarding the cell shape and size. So the cells in the human body vary greatly in their sizes. So when compared to the blood cells, we have red blood cells, lymphocytes, macrophages, megakaryocytes, etc. So RBCs are the smallest among these and are about 8 to 6 to 8 microns in size, and their function is to transport oxygen via hemoglobin. So they are of this size so that they can even enter into the small capillaries. Lymphocytes are slightly larger than the red blood cells. They are around 7 to 15 microns in size, and they are immune cells, and they have immune response with B cells and which are B cells and T cells. So the B lymphocytes and the T lymphocytes. Then we have macrophages, which are even more larger. They are around 20 to 30 microns. They are phagocytic cells and APC cells. So their function is phagocytosis and antigen presentation. Then megakaryocytes, as the name suggests, they are large cells. They are around 100 microns in size and they produce platelets in the bone marrow. RBCs are biconcave, disc shaped, and they are among the smallest cells in the blood, optimized for gaseous exchange and capillary flow. So the lymphocytes are slightly larger. Macrophages are much bigger due to their phagocytic function, and the megakaryocytes are huge cells in the bone marrow for platelet production. So you can see this is the biconcave disc shape of the RBCs, and it is of around uh, 7.5 microns plus or minus 1, and the thickness is around 2.5 microns. Then next is regarding the FITC stain. So FITC is fluorescein isothiocyanate, and it is a fluorescent dye that is commonly used in immunofluorescence and flow cytometry and other fluorescent based assays. So it has any fluorescent dye or any uh, a reagent that is used for visualization has an excitation wavelength and an emission wavelength. So the excitation wavelength of FITC is 595 nanometers and the emission is the excitation is 495 nanometers and the emission is 519 nanometers. So it emits basically green light and it looks green and it absorbs blue light. So when FITC absorbs the blue light, that is excitation, its electrons are excited to a higher energy state, and as the electrons return to the ground state, it emits green light that is around 590 nanometers. So the visualization is via a bright green color which we see upon excitation. So this is the excitation wavelength, this is the emission wavelength, and the appearance under microscope is bright green fluorescence. So this is a confocal image of a cell stained with phytid phalloidin. So phalloidin stains the F-actin filaments, and this is DAPI, which stains the nucleus. So we can see how neatly the stain has been taken up and how nicely we can visualize the cells using such reagents. So FITC basically emits green fluorescence upon excitation. Yeah. So next is regarding propydium iodide. So propydium iodide is used in FACS or for cell cycle analysis or death, cell viability analysis. So it is basically a DNA intercalating fluorescent dye. Excitation is 530 nanometers and emission is 670 nanometers. So it has red fluorescence, and it is widely used in FACS, that is flow cytometry or flow cytometry associated cell sorting. So PI is uh, it basically binds to the double stranded DNA in the cells, that is, it intercalates between them. The fluorescence intensity correlates with the DNA content. So, more DNA present will have more PI incorporated into it, so its intensity increases, allowing differentiation of G0 G1 stage, S stage, and the G2 M stage of cell cycle. So, however, it has limitation that PI cannot penetrate live cells, and cells must be fixed and permeabilized before staining. So, you cannot basically do any live cell experiments with PI because it is not penetrable into the live cells. So we also have acridin orange, Texas red, FITC, and propylium iodide. So acridin orange can bind to DNA and RNA. It is often used in viability assays or to distinguish between RNA, DNA, and less common for precise cell cycle analysis. Texas red is a fluorophore. It is usually conjugated to antibodies and not for DNA quantification. Then we have FITC, which is also fluorophore and is commonly used for labeling antibodies, not for DNA content. Then we have propydium iodide. It is a DNA staining reagent for cell cycle and most widely used for FACS for cell cycle analysis. And it is most commonly used dye for cell cycle by FACS analysis. So this is about the PI. And then coming to the nitrogenous bases. So the nitrogenous bases in nucleotides are classified into two. We have already discussed this. We have the purines, pyrimidines. Purines are double ringed structures, so they have fused five-membered or six-membered rings. They have fused five and a six-membered ring, and we have adenine and guanine as example for the purines. Then we have pyrimidines, which are single six-membered rings. So they are cytosine, thymine, and uracil. So uracil is only present in RNA, and thymine is only present in DNA. Adenine, guanine, and cytosine are present in both DNA and RNA. So you can see the structures here. This is adenine, this is guanine, this is thymine, cytosine, uracil. So adenine is a six-membered ring fused with a five-membered ring. Guanine is also a six-membered ring fused with a five-membered ring. So guanine and adenine are six-membered rings having a five-membered ring fuse. So there are two ring structures, and pyrimidines are single ringed and are one six-membered ring. So this is adenine, guanine, cytosine. This should be thymine. Okay, so that was about the nucleotides, and now we come to the difference in the densities of the cellular components. So, the density here refers to as the mass per unit volume. So, among the biological macromolecules and structures, DNA is very dense. It is due to its compact structure, that is the double helical structure, and it also has high phosphate content. So DNA is most dense. Its density is around 1.7 gram per centimeter cube. Highly compact, rich in negatively charged phosphate backbone. Proteins are next, which is around 1.3 to 1.4 gram per centimeter cube, and the average globular protein density is around this range. Then coming to organelles, we have them ranging between 1.06 to 1.2, and it depends on the type of organelle, if it's mitochondria, nucleus, lysozyme, etc. And next we have the mammalian cells. So mammalian cells also have a similar density, that is around 1 to 0.5 uh, sorry 1.05 to 1.1 gram per centimeter cube. So it's mostly water. 70% of it is water, so the density is overall density is very low. So the reason why DNA is densest is because DNA contains a phosphate sugar backbone, and bases are stacked very closely together. It forms highly ordered structure, compact, and is in a double helical fold. Then its compact packing gives DNA a higher mass per unit volume than proteins or whole organelles. Next is regarding osmium tetroxide. So, osmium tetroxide is a heavy metal compound commonly used as staining and fixative agent in electron microscopy, especially for transmission electron microscopy and also for scanning electron microscopy. So, in the role of osmium tetroxide in electron microscopy is it's a fixative. It cross-links lipids, preserving cell membrane and ultrastructure. A staining agent is osmium. Is a heavy atom that scatters electrons efficiently, providing contrast in the electromicrograph images. And the targets of this is the lipid structures such as the plasma membrane, organelle membrane, mitochondria, EI, and myelin sheath. So we have a table about the techniques, that is the FACS. So FACS uses fluorescent dyes, not heavy metal stains, and northern blotting detects the RNA using labeled probes, not EM staining. And southern blotting is defects in DNA using labeled probes, and no EM staining is required here as well. And then we also have a stain fixative, its function, that is the osmium tetroxide, lipid fixation, and electron dense contrast. Structures are visualized, membranes, organelles, ultrastructure, everything is visualized with the help of osmium staining. So it's basically a staining agent used in electron microscopy. So, yeah. So the next is a problem about the normality of NaOH. So to calculate the quantity of NaOH needed for a given normality, we first need to know the formula of normality, that is gram equivalence of solute by volume of the solution. Gram equivalence is mass by equivalent weight. So from this formula we have to calculate the amount required, that is the weight or the mass of NaOH required to prepare the given normality solution. So the weight is therefore normality into equivalent weight into volume. So to determine the weight, NaOH molar mass is 40 grams per mole, and for acid base reaction 1 mole of NaOH provides 1 equivalent of OH minus. Therefore equivalent weight of NaOH is 40 grams, and the weight that is required is normality into equivalent weight into volume. So given was 0.025 normality, and the equivalent weight is 40, and the volume prepared is 1 liter. So for 1 liter we have to calculate 40 into 0.025. It is nothing but 1 gram. So 1 gram of NaOH is required to prepare 1 liter of 0.025 normal solution of NaOH. Next is about the gel mobility shift assay, also known as electrophoretic mobility shift assay, EMSA. So it is a technique used to study interactions between proteins and nucleic acids, DNA or RNA. The principle is DNA or RNA fragments are labeled radioactively or fluorescently and incubated with protein of interest. If the protein binds to the nucleic acid, it forms protein nucleic acid complex, and the protein that has slower mobility is in a non-denaturing polyacrylamide gel compared to the free nucleic acid. So there is a shift in band. So the shifted band indicates binding. So it is used to detect specific DNA protein interactions, example transcription factor binding to promoters, study RNA binding proteins, quantify binding affinity, or test mutant proteins, DNA sequences, etc. So the complex studied is protein nucleic acid. The gel type is non-denaturing page, and the detection is using probes like radioactive, fluorescent, or chemiluminescent labels. And observable result seen is a shifted band, which indicates the binding or interaction. So next is bases are classified as strong or weak depending on their ability to accept the protons or dissociate in water. So strong bases are NaOH, KOH and CSOH, that is sodium hydroxide, potassium hydroxide and cesium hydroxide. They completely dissociate in water, 100% OH ions, strong proton acceptors. What is a weak base? Weak bases partially react with water to form ammonium ions and hydroxide ions. So, ammonia is a weak base. Only a small fraction of the ammonia molecules are ionized in water, therefore, it's a weak base. So, we have NH3 plus H2, which gives NH4 minus, that is the ammonium ions, plus hydroxide ions. So sodium hydroxide, potassium hydroxide and cesium hydroxide are all strong bases. They completely dissociate in water. Ammonia, however, is a weak base because it dissociates partially in water. Next is regarding the air bubble test for column chromatography. So to check for air channels or air bubbles in a column chromatography, an analyte with a KD value of 1 should be used. Why this is correct? Because it depends on the KD value. So the distribution coefficient describes how an analyte distributes itself between the stationary phase, that is the gel matrix, and the mobile phase, that is the solvent flowing through the column. So the analyte behavior is based on this KD. So if KD is equal to 0, an analyte with KD equal to 0 is completely excluded from the pores of the stationary phase. It travels through the column in the mobile phase, exiting at the void volume. So next is if the KD is 1, however, an analyte with KD 1 can access all the pore volume of the stationary phase in addition to the mobile phase volume. It is small enough to enter all the beads and travels the maximum possible path length through the column. This analyte will elute with a volume equal to the void volume plus the inner volume. So if the KD is more than 1, an analyte will absorb onto the column matrix and is retained for even longer. So detecting the air channels or uneven packing creates path of least resistance, which some mobile phase will take and bypassing the stationary phase. A tracer analyte with KD equal to 1 is used as a testing reagent because it is expected to travel through the entirety of the column, that is the packed bed, resulting in predictable illusion volume. If air channels are present, the illusion volume and the peak shape will be different from the expected result, indicating a problem with the column packing. Therefore, selecting an analyte with a KD of 1 is the standard method of testing column packing efficiency and identifying the issues of air bubbles. Next is the phenol chloroform method. Excuse me, just a second. Yeah, so the next is the phenol chloroform method, which is a classic technique used to extract DNA from cells, and it is used for separating it from proteins and other cellular components. The principle is basically after cell lysis, the cell releases DNA, RNA, and proteins. The lysate is then mixed with phenol chloroform mixture, and upon centrifugation, we have two distinct phases, which is an aqueous phase and an organic phase. In between that, we have an interphase. So the aqueous phase is at the top layer, which consists of water, salts, polar molecules like DNA and RNA. The interface is the denatured proteins and it accumulates as a center layer between the aqueous and the organic phase. So the organic phase has the phenol, chloroform, lipids, etc., which are not required, and it also contains lipids and hydrophobic molecules. So DNA is polar due to its phosphate backbones and is soluble in the aqueous phase, and only this aqueous layer is carefully collected for further precipitation of DNA or RNA. Proteins are denatured by phenol and partition at the interface, while lipids dissolve in the organic phase and are present in the bottom layer. So the basic steps of isolation of DNA RNA are lyse the cells and this releases the nucleic acids. We add phenol chloroform and mix it and then centrifuge it, which separates the phases. Phase separation occurs, and then we collect the top aqueous phase very carefully, which contains the DNA, and then it is precipitated using ethanol or isopropanol. So DNA is present in the aqueous top layer. RNA is also present in the aqueous top layer because both of them have the phosphate backbone, which makes them polar. And proteins are present in the interface. They are denatured, and lipids are present in the organic phase in the bottom. DNA is basically in the aqueous phase of the in the phenol chloroform extraction method. So I guess this is it. So we have discussed all the questions and topics for week 12. There was also an extra session last week, but nobody has attended it. However, I will record the session and upload it on YouTube, and the presentations will also be available in the NPTEL group, and it will be shared with you all. So that is it for this session. So all the best for the exams. I think the exam pattern has been shared with everybody. So hope you all do well. Thank you. So I think only one student is there. So if you have any questions, you can ask me. If not, I think we can end the session. I'll just wait for a couple of minutes. If you have any questions or doubts, you can just ask me. Okay, so no doubts. So thank you for attending. All the best for your exam. Do well. We shall end the session right now.